thank, I've had these folks for almost 2 years now. Ms. Samia, Ms. Dianna, and Ms. Shelby, and they're all really superb students, very active and engaged. A couple of them, God, I don't know how many recommendation letters I was writing for summer research internships at major research units. Shelby spent last summer at one already, and I think 15 letters or something, something like that. I forgot, I lost count. But at any rate, they're great folks. So a little bit more on histone mitochondrial interactions. So I leave it to you folks.
All right. Well, first I'll start off. My name is Shelby Harris. This is Dianna Thompson, and that's Samia Meera. And we are focusing on the study of the conservation of histone H3 binding to mitochondria using a yeast and cauliflower model. So would you like to start us off, Samia?
I would love to. All right, so I see there's a lot of students here and a lot of students that probably don't know what we're doing. So I'm going to put it in easy words so everybody can understand and we're on the same page. So first of all, our research is about the conservation of histone H3 binding to mitochondria, as we know that histones are a big part of packing up our chromosomes and our DNA, and which gives us our big chromosome structure. You know, the famous X structure that we always see on Google, which everybody knows about, right? All right, so that's what we're talking about, which this research is a lot zoomed in. So, you know, I will kind of come back and, you know, break it down for everybody that can understand it. All right, so first thing, we know that mammalian histones, which are a big part of our famous beads on a string structure, bind to the mitochondria and cause the release of cytochrome C, which can later lead on to programmed cell death, which cell death mechanism is very important in every cell because, you know, you cannot have cells just go on and divide and live their life forever, because what can that lead up to?
What I got.
Cancer. So we want cells to live a good, prosperous life and then end. And, you know, so over here we are using cauliflowers and yeast mitochondria. And so over here, histones are small, highly basic proteins which can easily stick to the negative charged DNA. Histone H3 is one of the core histones that help provide the chromatin structures in eukaryotic cells. All right, so we have histones are highly modified post-translationally, and they lead up to programmed cell death mechanism. And it is an intracellular program, which means that our cell death is a very clean process when cells just decide to pack up and, you know, get in their life and go. All right, so abnormal regulation of programmed cell death can cause various diseases, which we already know about, like I've mentioned here, developmental disorders, neurodegeneration, and cancers, which we do not want to happen. So Dianna will now help us understand all the methods that we are using to get to our stuff.
So we use cauliflower and yeast to get the mitochondria from the cauliflower and the mitochondria from the histone. That was not right. I'm sorry. So we have cauliflower, and we get the mitochondria and histones from the cauliflower. We get the yeast, and we get the mitochondria and the histones from the yeast. So from the yeast, we grow up to a certain range so that we have nice cells. And then from there, we take them and we lyse the cell wall with a ligocase solution. And then from there, we homogenize the cells, meaning we're going to break them up, and then we're going to put them in a centrifuge, which means we're going to spin them down really well, and then we're going to have a pellet that has a lot of like full cells and like concentrated cells, and then we'll have the supernatant that is just mostly broken up cells, and with that, we take the supernatant and we use a Perkal density gradient to get the mitochondria through, so we can get purified mitochondria from that. So, and then we use, to get the histones, we take the pellet that we got originally and we use an epigenetic histone kit to get out the histones. And we do the same thing to the cauliflower, but instead we grind the cauliflower into thin sheets of paper, or thin sheets of cells, like paper. And then We also use the Percol gradient to get the mitochondria, and we use the epigenetic kit for the histones as well. And we did cross incubation to determine if the the mitochondria from the cauliflower bind to the histones from the yeast, and then other things vice versa. And we also use mammalian histones as like the other type of histones that we use. And we did it as our normal, which would be a 2 molar salt concentration, and we also did it with higher salt concentrations, so to determine if the binding would be disrupted by the high salt concentration. And from there, we used Western blotting and SDS page to analyze the results.
So starting up here with this first blot, we have our yeast salt resistance. And like Diana said, the salt resistance is to test how strong these bonds are between these and how long we can take before we pull off the histones from the mitochondria. So starting out over here, we have our molecular weight standard to tell us where it is. We have our control. The control is there to tell us where we should be looking and to check our antibodies. Right here in these two lanes, there's nothing there, which is good. If you come up here, you can see a very, very light band here, and that's because our histones are in the supernate where they should be, versus they're on the mitochondria, which is in this lane. And since there's no histones here, none of our antibodies stuck there, so they showed no signs, so we know that we have no contamination in our control. Here we have what's called the incubation step, where we add in our histones to our mitochondrial sample, and as we can see, we have histones binding now to our mitochondria as we have a band showing up. But we have very little in the supernate, if not none. Over here we have a respin step, and a respin step is a secondary control to test to see if we didn't have any mitochondrial lysing. So if the mitochondria would have broke apart and degraded, we would have the release of histones into the supernate. But since we don't have any bands here, we would have these bands, and it shows that our mitochondria are still intact and histones are still binding to them. And then over here we have where we went up to 1.4 salt molarity, and we still have our histones binding to our mitochondria given by a band. So we said that up to 1.4 salt concentration, the mitochondria still stay on. They still have those strong forces in between them holding them. And then we went up to 6. And as you can see here, as we have lighter bands, we said that, well, at 0.6, they have started to come off, but they're still strong enough to stay on the mitochondria. So we know that it's resistant to very, very high salt concentration. And the second one we have the cauliflower salt resistance, which is the exact same as the yeast, except for here we have 2 bands, and this is because cauliflower, like Diana said, we have to grind it and we have to blend it. And when we grind it and blend it, we get a lot of cells premature lysing. So we have a lot of histones and mitochondria already released into our sample before we can ever do our project. So that's why we have 2 bands here, because we already have histones in our supernate and histones attached to our mitochondrial pellet. But when we incubate, meaning when we take our mitochondria sample and we add histones to it, we see that the histones are leaving the supernate and going to the mitochondria. So we know that they're choosing to go to binding to the mitochondria than staying in the supernate. In our next step, we have up to 4.1 salt, so we went straight into the salt resistance. At 1.4 molarity salt, we have binding still with no in the supernate. At 0.6 salt, we still have really strong binding. Really dark bands, really light bands in the supernate. At 8.8, slightly lighter, but still pretty dark. So we know that cauliflower mitochondrial samples can hold up to 0.8 molarity salt, which is pretty high. So we know that they're holding up too with strong bonds. And the next step, we did the cross incubation, like Diana said, where we took yeast mitochondria and cauliflower histones, and we said, well, would these 2 species stick together? And the answer is, they did. So we have our control pellet, so we have empty lanes. There's no contamination here because we use yeast. But here, when we added in our mitochondria, they went to the pellet. They went to the mitochondria. They chose to leave the supernate and go to the mitochondria and bind. Same thing here. We have 0.4 salt, still binding, very little in the supernate. At 0.6, it's kind of the same color, so they're still holding up at 0.6. You can kind of say there's some here, but it can't really tell it's really light. And then here at 0.8, they're still binded. They're still on there. So they're holding up to 0.8 too. So we say that this is very strong. This is highly conserved because when we took it from the yeast and we took it from the cauliflower and we put them together, they did the exact same thing as they did with each other. And so we said, well, what if we use mammalian histone? So yeast and cauliflower, we have that, but what if we do yeast and animals? And we said, well, will it do the same thing? And the answer is, yeah, it did. So we have our empty bands here with no contamination to control. We have the pellet, the mitochondrial pellet, with the histones leaving the supernate to bind. And then we have it here at 0.4 molarity salt, still binded, still on there, they're still holding on. Up to 0.6, they're still on there. At 0.8, it got a little bit lighter, and you can see faintly a band here. So up to 0.8, you can kind of say that they're kind of coming off, but they're still resistant to this very high salt. So what we got from this is that this is very, very strong. This is something that's happening, and it's not something that's just going to happen for a second and not do anything. This is staying on there, and that leads us to believe that this might have a causation for something, say like apoptosis or anything else. And that it's conserved. So this is something that happens often. If all the cells are doing it, whether their species is, it's something that probably has a purpose, or else evolution wouldn't keep it in. So now we move on to our conclusion.
So I'm going to summarize up what she said. So, so under regular incubation studies, they like to bind together, and then under low salt concentrations, the mitochondria and the histones bind, and once it gets higher, we have a progressive decrease in the amount of how much is binded together, but it's still binding, so we still have a reason to believe that this is a very highly conserved mechanism through all of the eukarya. And so future studies will involve purifying individual histones with HPLC, and we'll test each one for mitochondrial binding and for the release of cytochrome C.
All right, so wrapping up everything, this was our biggest thing. We wanted to see that this mechanism is happening in every single cell type. It must be important. What is going on that is causing this cytochrome C release? And, going back to this cell death mechanism, this, it is connected to so many disorders, so many diseases, cancer, stuff like that. So our biggest thing in, down the road, in the future, when we all are researchers and have our PhDs and stuff like that, hopefully gonna be continuing to work with this, because we want to see what is happening that is not going on in cancer cells, that they're not dying and stuff like that. What is the normal process that we are trying to find? We have this one big picture, and we are taking these small puzzle pieces that we have been working on for 2 years now, and later on we will have more pieces to join together so we can bring back. And this is our small little effort to understand what we can fix to help people, clinically in the hospitals, in the research field. What can we do? So this is kind of our contribution to the world, to the medical field, pretty much. And I hope you guys really enjoyed this. If you have any questions, please let us know.